All right. Hello, everyone. Welcome back to my channel. My name is Dr. Anwar Yusuf Dunbar, as you know, and this is Big Discussions 76, Science and Technology. First of all, please like this video, please share it, and please subscribe to my channel. If you want to donate something to the channel, that information is below in the description box. I don't have AdSense over here or Super Chat at this point. Creating science content is a little tricky, so it might be a little while before I have those over here, but I think at some point I will. Also, please consider joining the Big Words LLC Newsletter. In addition to being a YouTube content creator, I am a writer with two blogs and a book project on the way. Well, you'll notice some changes here. I have realized that I can use a StreamYard to make pre-records. And this is particularly useful when, in this instance, I have an article that I want to share and I can share it with you all. I'm going to read it. I'm going to show some diagrams here, but this is one of my first times doing this. So you don't have to stream live to use StreamYard. So I recently became a member of AAAS, the American Association for the Advancement of Science. That is and I had reasons for joining that society, which I won't get into now, but I have this YouTube channel and I write, so I've become more and more interested in science communication, science writing, science video, the communication of science. So when you join AAAS, depending upon which package you purchase, you also get a subscription to their science magazine. And there are numerous science articles in their magazine. And I believe I'm still new, the magazine comes out weekly. So there's a lot of science content and information that they share with their members. And I just sat down and browsed through it for the first time this weekend. And a article that I thought that was worth presenting. Well, this video is going to be based upon an article that I thought was worth presenting that I saw in their magazine. So, we're going to do a little bit of cell biology, and we're going to talk a little bit about cancer, and we're going to talk a little bit about, we're going to dig CRISPR back up. Let me see if I can remember how to say it. CRISPR is an acronym, and the first time I talked about CRISPR on this channel was with Nicole Ali. We haven't spoken in a while, but CRISPR is an acronym for Clustered Repeated Interspaced Palindromic Repeats. So I still remember the acronym. Um, so CRISPR is gonna come up in this summary. So without further ado, and I think it's okay for me to share this. I mean, I did purchase the membership, and so I do have access to this information. So if anyone from AAAS is seeing this and I'm not supposed to be sharing this, let me know. But in the meantime, I have access to it so I can share it with my audience. I am advancing science. I am communicating science. So there are going to be some words in here some of you may not understand. I will do my best to distill out the meanings and make it so that you can understand what's being discussed. There's a nice pretty diagram slash cartoon at the end of this. So I think this should be pretty easy to follow along. And this is just a summary. There's a full article somewhere with all the methods and all the figures and legends and tables. But the whole point of this is to make it understandable understandable by the masses for the science community and people in different segments of the science community. So I'm going to pull up the article and I will share it and we'll go through it. So this is the actual cover of this particular issue of Science magazine, and that is a depiction of the Golgi apparatus or the Golgi bodies. And going back to cell biology 101, when you think about the Golgi bodies, you're thinking about packaging and trafficking. So you're creating something within the cell, you're packaging it just like the post office or UPS or FedEx, and then you're shipping it off to other locations within the cell or to other cells in the organism. And you're typically shipping off some sort of protein. So we're going to get to that. That's a pretty picture. It caught my eye when I saw it. So let's go to the article of interest. Uh-oh. Let's go back. All right. And it turns out this is the article that I wanted to focus on. So this one, it's dated October 7th, 2022, and it is entitled Lysosomal Enzyme Trafficking Factor, LYSET, Enables Nutritional Usage of Extracellular Proteins. So lysosomes, lysosomes. So within animal cells, there are proteases, there are protease systems. In my graduate research, we focus on the 26S proteasome, but there are also lysosomes within our animal cells, which are important for cleaving proteins. I believe it's proteins. I believe it's proteins, but there are different types of proteases and protease systems, and there's a trafficking factor that's discussed in this article called LYSET, and that's an acronym. I believe that's an acronym for something. But let's take a look. So the introduction reads: Mammalian cells are surrounded by a range of different nutrients, including amino acids and extracellular proteins. In nutrient-rich conditions, cells prefer to import free amino acids to meet their nutritional demands. However, most amino acids in circulation and in extracellular space are contained within proteins. If you go back to Cell Biology 101, the basic building blocks of proteins are amino acids. Cells can ingest proteins from the environment and deliver them to lysosomes, organelles with digestive enzymes that break down proteins. I'm sorry, organelles with digestive enzymes that break proteins down into their constituent amino acids. Okay. By generating an intracellular nutrient source, lysosomes can sustain cellular functions during starvation. This process is commonly exploited by cancer cells, which can feed on extracellular proteins to thrive in poorly vascularized, nutrient poor tumors. However, the molecular pathways that enable cells to use extracellular proteins as a nutrient source remain completely understood. So that's one of the features of cancer cells and that's that they're able to survive and thrive in environments, for example, in this case, where there's no circulation. So they're able to um, acquire the nutrients and the food sources that they need in abnormal environments. Our cells need oxygen and they need to be able to release carbon dioxide, and that requires close proximity to our vasculature. But cancer cells can survive in a little further away from the vasculature. So, in terms of the rationale, we set out to identify genes that are essential for survival and growth when cells are rely on extracellular proteins as nutrients. Genetic screens have been instrumental in functionally characterizing genes in mammalian cells and identifying genes that become essential in specific cancer contexts. However, such screens have commonly been performed in cell culture media that provide most amino acids as supraphysiological levels while being strongly depleted in extracellular proteins. Conceivably, such unphysiological nutrient mixtures enforce metabolic activities that differ from in vivo phenotypes. To address this, we developed screening conditions where cancer cells grow either by the import of free amino acids or by the uptake and lysosomal degradation of extracellular proteins. So in terms of the results, and there's a pretty cartoon at the end of this that shows us what all of this means. But let's just get through the text first, and then I'll show the cartoon and break it down. So in terms of the results, through CRISPR screens in defined nutrient environments, we identified LYSET, a transmembrane protein selectively required for cell survival and growth when extracellular proteins were an obligatory amino acid source. So when extracellular proteins are the only food source, this lyset protein is necessary. I don't quite know how they used CRISPR here, but CRISPR was used in these experiments. Mechanistically, we characterized lyset as a protein that anchors GLICNAC1 phosphotransferase in Golgi membranes for tagging catabolic enzymes with the lysosomal trafficking signal mannose 6 phosphate. GLICNAC1 phosphotransferase stability depended on lyset because of its transmembrane domain, which was found to contain multiple hydrophilic amino acid residues and to co-occur with lyset in multicellular animals. Without lyset, the GLICNAC1 transferase transmembrane domain was unstable, which led to degradation of the protein. Okay, so I'm going to stop there. Okay, so within our cells, um, you have proteins that are that perform certain functions, and then you have other proteins that are necessary for the proper orientation and domain architecture of those other proteins. So this is saying that without the lyset, the GLICNAC 1 phosphatase protein, it can't orient itself, or it doesn't become properly oriented, and it can't perform its function. So the GLICNAC 1 phosphatase protein, it needs the lyset protein to get set up and to perform its function. I'm trying to distill down what that all means. I'm sorry, and it's GLICNAC 1 phosphotransferase. When you're dealing with cell biology and enzymology, the names get pretty long. So it's GLICNAC 1 phosphotransferase. Okay. Now here's another interesting point. When proteins, and I worked on this during my own graduate research, when proteins are synthesized and they're misfolded, and when they aggregate and when they can't perform their function, oftentimes they immediately get triaged or segued for degradation within the cells. Okay. All right. Consequently, catabolic enzymes did not reach the lysosome and were instead mistrafficked to the cell surface. Lyset deficient cells were unable to generate nutrients through lysosomal breakdown of proteins and accumulated lysosomes that were filled with undigested cargo. Although lyset deficient cancer cells grew normally under nutrient-rich conditions, they failed to grow in amino acid poor environments and displayed a severely reduced ability to form tumors in mice. Okay. Let me see if this blows up. Okay. I'm going to try to blow this up and I hope that works. Hope you all are able to see that better. Okay, so over on the right, you have the wild type. That's the normal cell. You see amino acids going in through those channels into the cytosol. The cell is ingesting proteins from from its exterior matrix. Those proteins are going into that lysosome where they're being cleaved by the protease, the mannose 6-phosphate. So in the Golgi apparatus, you see the lyset and you see the GLICNAC1 phosphotransporase, and that GLICNAC1 phosphotransporase is tagging that um, that protease with the phosphate sugar there. So once that gets tagged with the phosphate sugar, it goes into the lysosome and it cleaves the protein into amino acids that the cell can use for its own metabolism. But once it looks like they used CRISPR to eliminate the lyset, and once you eliminate the lyset, the GLICNAC1 phosphotransporase gets trapped in the Golgi apparatus. So once that gets trapped in the Golgi apparatus, it can't tag that protease with the sugar, the phosphate sugar, and so that will not go into the lysosome to cleave those proteins, and they stay in the lysosome and they build up. That leads to cell death because the cell basically can't eat. So, as they said in the text, in the nutrient-rich environment, these cancer cells that are lyset deficient, they can eat, they can ingest amino acids, but when you put them in a media that's lacking free amino acids and other food sources, they basically starve and they die. Okay, I hope that was clear. Let's see. And this legend says right here, lyset anchors GLICNAC1 phosphotransporase in the Golgi for tagging catabolic enzymes with the lysosomal trafficking signal, mannose 6-phosphate. Okay, the generation of catabolically active lysosomes enables cells to acquire amino acids through the breakdown of extracellular proteins. Loss of lyset abrogates the mannose 6-phosphate pathway, depletes the lysosomes of catabolic enzymes, and blocks the survival and growth of cells that rely on extracellular proteins as nutrients. Okay, we're almost done here. Our results identified lyset as a core component of the mannose 6-phosphate pathway for lysosomal enzyme trafficking. A clue for the function of lyset came from our discovery that GLICNAC1 phosphotransporase contains an energetically unfavorable transmembrane domain, which was predicted to depend on lyset for stable membrane integration. The co-occurrence of lyset and the unstable GLICNAC1 phosphotransporase transmembrane domain in the same organisms suggests that they became functionally linked during evolution of the mannose 6-phosphate pathway. Conceivably, controlling GLICNAC1 phosphotransporase levels through lyset constitutes a mechanism to regulate lysosomal enzyme trafficking. Lyset is relevant for several human pathologies. Lyset deficient cells lack a functional mannose 6-phosphate pathway, which provides a mechanistic explanation for the onset of lyset mutations with hereditary syndromes that resemble the lysosomal storage disorders of mucolipidosis 1, I'm sorry, mucolipidosis 2 and 3. Moreover, lyset enables cancer cells to exploit extracellular proteins as a nutrient source, thereby gaining metabolic flexibility and resilience, thus inhibiting lyset and the lysosomal enzyme trafficking pathway might be a promising strategy to suppress a key metabolic adaptation in cancer. Okay, well, I don't know about you all, but I feel a little bit smarter after reading that. I did not acknowledge the authors of this. Anytime you're presenting something, you want to provide proper attribution. And I think in this instance, since this is coming from the Science Journal, and this is my first time doing this, I need to give credit where it's due and provide attribution and um, at least acknowledge some of the authors. So it looks like Katerina uh, Pachincha is the first author on this paper. I'll read the first three. So Katerina Pachincha, Sven Grossel, and Robert Callist. Those are the first three authors. And most likely, Wilhelm Palm is the principal investigator. And, well, Marius K. Lemberg, Johannes Zuber, and Wilhelm Palm. At least one of those individuals is probably the principal investigator, though there may have been multiple labs who contributed to this. So there's a lot there. This was a mechanistic cell biology study, but also it gave potential indicators as to how cancer cells survive and thrive. And they're proposing that is through this LYSET protein, which allows proper um, trafficking of the protein GLICNAC 1 phosphotransferase, which tags proteases with the mannose, a sugar phosphate. I'll just say a sugar phosphate. So I'm going to stop this here. Um, I want to applaud those of you in my audience if you followed along to the end. This is what it's like talking science. This is what at least scientists in the biological sciences and in the biomedical sciences, this is how we talk amongst each other. This is how we talk about data. This is how we present results to each other. And this is what we do. So this is why it's important to be able to read, and it's important to be able to exchange ideas and communicate articulately with our peers. So I'm going to stop this here. So this is not below in the description box because I don't think if you don't have access to this, you probably can't read this, but maybe you may be able to look this up on PubMed or in Google yourself. So in any case, thank you for watching. Please like, share, and I'm sorry. Please like, share, and subscribe. If you liked this format, let me know. I will at least do a few more of these. And as I see more interesting articles in the AAA spaces, yeah, I will share them with my audience. But let me know if you liked this. Let me know if you have any questions. Let me know if there's a particular part of this that you understood or that you didn't understand. Let me know. Please like, share, and subscribe. If you want to donate to the channel, that information is below in the description box. And also, please consider joining the Big Words LLC Newsletter. Everyone stay safe out there. And as always, remember that your attitude determines your altitude. Take care, and I will talk to you the next time. Bye-bye.